Hello students, welcome. Myself Bimlendu, working as assistant professor at IIMT College of Pharmacy. In today's lecture, we will start our biochemistry with chapter 1, which is the introduction to biochemistry. This is the syllabus. This is the this is the syllabus as per the EP Board of Technical Education, starting with the definition of biochemistry. Biochemistry can be defined as the structure. Biochemistry can be defined as the study of structure, composition, and chemistry of and chemical reactions of molecule in living system. The term biochemistry was first introduced by the German chemist Carl Neuberg in the year 1903. Biochemistry may be defined as the science of chemistry of living matter in its different phases of activity. Biochemistry, in its broad aspect, the most comprehensive of all the branches of the chemistry, includes organic, inorganic, and physical chemistry to the extent it is related to the chemistry of living things, both the plants and animals. We talk about the objectives of biochemistry. The main objectives of biochemistry are to study and understand the structure and properties of substances constituting the framework of cells and tissue. Second one is to study the structures and properties of substances which enter the cells as source of energy or levels or leave the cells as waste products. To study the catalytic activity of enzymes. To study the process that convert diet into compounds which are characteristics of the cells of a given species. To study the manifold energy requiring process of the living cells. To study the chemistry of inheritance. Now, next topic is the scope of biochemistry. The main scopes of biochemistry are the study of cell structure and its components, chemistry of carbohydrate, protein, lipids, and amino acids, chemistry of inorganic elements and their deficiency, a study of enzymes, water metabolism, their sources, regulation, etc., a study of vitamins and their deficiency, immunochemistry, is a deficiency of a study of immune system, metabolism of carbohydrate and their metabolic disorder, metabolism of protein and amino acid and their metabolic disorders, metabolism of lipids and their metabolic disorder, chemistry of nucleic acid, recombinant DNA technologies, nucleoproteins and metabolism of nucleic acid, detoxification mechanism, hormones and their biochemical role in the body. Applications of biochemistry in pharmacy. The main applications of biochemistry in pharmacy are drug constitution. Biochemist helps in the constitution of new drugs. The half-life. The half-life of a drug is the time it takes for a drug's active substance in our body to reduce by half. Biochemistry helps to determine the half-life of a drug. Third application is chances of degradation of drug. Biochemistry helps to determine the stability of a drug by calculating the chances of its degradation with changes in atmospheric conditions like temperature, humidity, etc. Fourth application is the expiry date. Determination of biochemical test helps to fix the expiry date of the drugs. Drug storage. The storage condition required for a drug can be determined by the biochemical tests. Drug metabolism. Biochemistry gives an idea of how drug molecules are metabolized by different biochemical reactions in the presence of enzymes. This helps to avoid drugs that have a poor metabolism. Next application is it detects nutritional deficiencies. Deficiencies of biomolecules such as proteins, lipids, vitamins, minerals, and water may cause diseases. Biochemical research has a role in preventive, preventive medicine by helping identify such diseases. Disease understanding. Health depends on the proper balance of biochemical reactions occurring within the body. Abnormalities in biochemical reactions result in diseases. Biochemistry helps to understand the diseases of protein, energy malnutrition, involve error in the metabolism of carbohydrate, proteins, lipids, etc. Diagnostic biochemical test helps to understand the diseases and the necessary treatments. Coming to the next topic, which is the cell and its biochemical organization. Cell which form the tissue, and tissue form the organism. Organism make up the organ system, and organs finally make up the living body. Starting with the cell, definition of the cell. Cell is defined as the basic living structure and functional unit of an organism, which is capable of carrying out the process of life independently. Cells are the basic building block of life. Cells it was discovered by Robert Hooke in 1665. Its cells can be prokaryotic or eukaryotic. Prokaryotes include bacteria and lack a nucleus or membrane bond structure called organelles. Eukaryotes include other cells, have a nucleus and membrane bond organelles. Examples are plant, fungi, and animal cells. This is the difference between prokaryotes and eukaryotes. A type of cell present in the prokaryote is always unicellular. In eukaryote, it is unicellular and multicellular. Cell size. In prokaryote, cell size ranges from 0.2 micrometer to 2 micrometer in diameter, whereas in eukaryote, cell size ranges from 10 micrometer to 100 micrometer in diameter. Nucleus is absent in prokaryote, whereas it is present in eukaryote. Ribosomes, these are present in both prokaryote and eukaryote, but it is smaller in the case of prokaryote, smaller in size, and in the case of eukaryote, it is larger in size. Shape is also different. In prokaryote, the shape is spherical, and in eukaryote, it is linear in shape. DNA arrangement in case of prokaryote is circular, whereas in case of eukaryote, it is linear. Mitochondria is present in prokaryote, and it is absent. So mitochondria is absent in the case of prokaryote, and it is present in the case of eukaryote. Cytoplasm is present, but cell organelle is absent in the case of prokaryote, and cytoplasm is absent, and cell organelles are present in the case of eukaryotes. Endoplasmic reticulum is absent in the case of prokaryote and present in the case of eukaryotes. Ribosomes, these are smaller in the case of prokaryotes and larger in the case of eukaryotes. Reproduction, prokaryotes grow by the process of asexual reproduction, where the eukaryotes follow both asexual and sexual reproduction. Examples of prokaryotes are bacteria, and eukaryotes are plant and animal cells. Structure of prokaryotic cell. This is the structure of prokaryotic cell. It's comprised of different parts. We will discuss this all the parts one by one. This is the basic intro of the prokaryotic cell. Prokaryotic cell does not have a nuclear membrane. However, the genetic material is present in the region called cytoplasm, as known as the nuclear. Let us discuss one by one with these parts of prokaryotic cell. Capsule, outer covering that protects the cell when it is engulfed by other mechanism, assist in assists in retaining moisture and helps the cell to adhere to the surface of the nutrients. Cell wall. The cell wall is an outer covering that protects the bacterial cell and gives it shapes. Cytoplasm. Cytoplasm is a gel-like substance composed of mainly of water and that also contains enzymes, salts, cell component, and various organic molecules. Cell membrane, or which is also known as plasma membrane. The cell membrane surrounds the cell cytoplasm and regulates the flow of substances in and out of the cell. Pili or pilus, which is a singular form. It is a hair-like structure on the surface of the cell that attach to the other bacterial cell. Shorter pili are called fimbriae. It helps bacteria to attach to surfaces. Flagella. Flagella are long, whip-like protrusions. Pro, Flagella are long, whip-like protrusions that aid in the cellular locomotion. It helps in the cellular locomotion. Ribosomes are cell structures responsible for protein production. Plasmids are gene-carrying circular DNA structures that are not involved in reproduction. Nucleoid region, area of cytoplasm that contains a single bacterial DNA molecule. Second type of cell is the eukaryotic cell. This is the structure of the eukaryotic cell. We will study these all the parts one by one. First part is the cell membrane or plasma membrane. Singer and Nicholas, these are the two scientists who proposed the fluid mosaic model of the cell membrane in 1972. The cell is separated from its surrounding by a thin membrane called cell membrane or plasma membrane. Cell membrane is formed of 40% lipid and 60% protein. The cell membrane is a double layer of phospholipid molecules. Proteins in the cell membrane provide structural support from channels for the passage of material that act as receptor sites. Function of a cell membrane. The main function of membrane are it holds a cell, it serves as a barrier to the outside and permits only selected compounds such as nutrients to pass through. It secretes waste product, it keeps out toxic material, it prevents the loss of essential substances from the cell, it protects the inner part of the cell. Second part is the nucleus. Within the cell, there is a central mass called the nucleus, which is surrounded by a double-layered nuclear membrane called the nuclear envelope. It is more or less spherical in shape. The inner membrane joins at intervals to form pores. From pores, the exchange of material takes place. The living matter inside the nuclear membrane is called the nucleoplasm. The nucleoplasm also contains the nucleolus, which contains RNA, DNA, and other genetic material. The major part of the nucleoplasm is occupied by a thread-like structure called chromatin, which consists of deoxyribonucleic acid. Functions of nucleus. The nucleus controls all functional activities of the cell. The nucleus contains the most important component called chromatin. Before cell division, chromatin organizes themselves in a small, simple thread-like structure called chromosomes. These chromosomes are equally distributed to daughter cells. After cell division, chromosomes get converted back to the chromatin by uncoiling. It is the site of DNA-directed RNA synthesis, which is involved in the protein synthesis. Third part is the mitochondria, which is known as the powerhouse of the cell. Mitochondria are rod-like structure and the largest component of the cytoplasm of a cell. Each cell contains 50 to 2500 mitochondria. A mitochondria consists of proteins, lipids, and small amounts of nucleic acids. It is composed of double membrane system. The outer one is smooth and covers the organelle. The inner one is folded and forms cristae. Energy producing chemical reaction takes place in the cristae. The internal chamber is referred to as the matrix of mitosol. This matrix contains several enzymes, circular double-stranded DNA and RNA and ribosomes. This is the structure of mitochondria. Functions of mitochondria. It plays an important role in respiration and energy production. It is responsible for the transportation of chemical energy into biological energy in the form of ATP. Mitochondria is responsible for the transmission of heredity characteristics. It contains enzymes that control the synthesis of lipids. Fourth part is the endoplasmic reticulum. Within the cytoplasm of the cell, layers of the cytoplasmic membrane are folded inwards, forming vesicles close to the surface of the cell. This complex within the cell is called endoplasmic reticulum. This is a diagram of the endoplasmic reticulum. Endoplasmic reticulum is of two types. First is the rough endoplasmic reticulum, which contains the ribosomes that are responsible for protein synthesis. It is also called the granular endoplasmic reticulum. Second one is the smooth endoplasmic reticulum, which is agranular endoplasmic reticulum. These do not contain ribosomes and are responsible for fatty acid and steroid synthesis. They also store and release calcium. Functions of endoplasmic reticulum are rough endoplasmic reticulum is involved in the protein synthesis, while the smooth endoplasmic reticulum is concerned with the lipid and glycogen synthesis. It gives mechanical support by forming skeletal networks. Include spaces of the endoplasmic reticulum play a role in exchange of material between the cells and the extracellular fluids. These are the enclosed spaces. The exchange of material takes place by the process of diffusion, active transport, phagocytosis, or pinocytosis. Next part is the Golgi body. Golgi apparatus, it is also known as Golgi apparatus, is the membrane-bound organelle of the cells that is made up of series of pouches called cisternae. This is cisternae. In the diagram of Golgi body, we can see these are known as cisternae. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to target destinations. It is located in the cytoplasm next to the endoplasmic reticulum and near the cell nucleus. Functions of Golgi bodies are: it is a site for enzyme secretion and it participates in the formation of lysosomes. Golgi apparatus is also involved in membrane synthesis, particularly for the formation of intracellular organelle. Next part is a lysosome. It is a membrane-bound organelle that is responsible for the degrading protein and membranes in the cell, and also helps to degrade materials ingested by the cell. Next part is cytoplasm. It is the cellular matrix. The cellular matrix is collectively referred to as cytosol. The cellular matrix which are present in the cell, they are collectively known as the cytosol. The cytosol is basically a compartment containing several enzymes, metabolites, and salt in an aqueous gel-like medium. Within the cell, there is a cytoplasm containing organelles embedded in it and soluble component. The soluble portion of cytoplasm is termed as cytosol. Function of cytoplasm: it helps in maintaining the osmotic pressure inside the cell. These are all the parts of we have covered the prokaryotes and eukaryotes. They are all parts. Thank you, students.